All right. This is our group presentation for the Bio 440 class. The presentation, Comparative Genomics of Antibiotic Resistant Genes in E. coli O157H7 using BLAST. Our group consists of Sean Snyder, Jonathan Malan, Jalen, and Hamza. The abstract is the increasing resistance of E. coli bacteria to antibiotic treatments fuels a growing threat to health globally. This study employs BLAST-based comparative genomics methods to investigate resistance gene distribution in commensal as well as pathogenic strains of E. coli through computational analysis with emphasis on E. coli O157H7. This investigation aims to analyze both genetic sequences and evolutionary mechanisms that affect resistance genes. Lab findings reveal the unique resistance characteristics of individual strains together with the natural capability of crucial resistance factors to move between genetic elements. Now the introduction. Antibiotic resistance develops when the organism evolves defense mechanisms to better survive the impacts of antibiotic treatments. Pathogenic strains such as E. coli O157H7 are a highly prevalent model organism used extensively for bacterial and antibiotic research. We examined this bacterial strain due to its vast scientific relevance as a model organism, as well as for the thorough background research already prevalent on the topic. The plethora of research on this strain is not without warrant, as each year in the United States, E. coli infections cause approximately 265,000 illnesses and about 100 deaths every year. And approximately 40% of these infections are caused by the strain E. coli O157H7. If this development of antibiotic resistance is left unchecked, future antibiotic tolerant bacteria may develop and spread at a faster pace than humans can develop antibiotics or circumnavigate. By identifying the genes responsible for antibiotic resistance, the search to decipher how and why these genes promote resistance can be unlocked to one day assist in promoting solutions to the problem of ever-advancing drug-resistant bacteria. Now I'm going to go ahead and pass it over to Sean.
So we hypothesized that with this research, the modern strains of antibiotic-resistant E. coli, such as O157H7, would share a common ancestor in which the antibiotic resistance and the genes originated. And our methodology to research this was to use NCBI's database as well as the National Database of Antibiotic Resistance Organisms, more specifically BLAST, BLAST-N, and the T-BLAST-N queries to compare the presence of known antibiotic resistance genes in the genomes of a variety of different E. coli strains. And through the utilization of these tools, we were able to compare the resistance genes between and across the E. coli strains, which allowed us to obtain E-values and percent identity measures, as well as alignment scores, which determine the match strength. Additionally, we used a phylogenetic tree to display the relationship between the different strains. And here, up in the top left, we can see a phylogenetic tree that shows Rep A and Fin O genes on the P0157 plasmid of E. coli O157H7. And it closely resembles those found in other bacteria like Salmonella, highlighting the cross-species genetic exchange. This gene-sharing mechanism aids bacteria's survival and significantly contributes to the spread of antibiotic resistance. And these findings support the idea that plasmid-driven gene movement plays a critical role in the rapid dissemination of antibiotic resistance across these bacterial species. Now I'm going to pass it to Jalen.
By using BLAST-N to blast the E. coli gene, antibiotic-resistant gene, BLAVEB, and using an MSA viewer, it shows that there are very few differences between the genes. This indicates high conserve rates between them and indicates that the gene is highly conserved. And by using T-BLAST-N with the same sequence and target, it shows that there is a high level of conservation between the proteins. And now I'm going to go to Hamza for the conclusion and results.
All right, in the results sections, when analyzing antibiotic-resistant genes in E. coli, we observed a clear distinction between commensal and pathogenic strains. Pathogenic strains like O157H7 carried significantly more ARGs, including key resistant genes like BLA, OXA, and SUL1, while commensal strains had little to no ARG expression, likely due to reduced exposure to antibiotics. Although we identified dozens of resistant genes, our BLAST comparison didn't show a single gene consistently present across all pathogenic strains. This shows how scattered and complex antibiotic resistance truly is. We initially hypothesized that these ARGs would track back to a common lineage, but our findings didn't support that. Instead, the data strongly point to horizontal gene transfer as the main driver behind the spread of resistance, explaining why antibiotic resistant genes appear across diverse and unrelated strains. In the conclusion, one of the main takeaways from our research is that E. coli acquires antibiotic resistance largely through horizontal gene transfer, not just between its own strains, but across different bacterial species. This gene sharing allows resistant traits to spread rapidly in diverse environments. We found a clear difference between commensal strains, which typically live in our gut without causing harm, and pathogenic strains like O15787. The commensal strains had little to no expression of resistant genes, while the O15787 showed high levels of antibiotic resistance, like genes like BLA, OXA, and SUL1, which are known to help bacteria survive antibiotic treatments. The reason behind this unequal distribution lies in the selective pressure. Bacteria exposed to higher levels of antibiotics are more likely to develop and retain these resistant traits. These findings highlight the importance of genomic surveillance. By using tools like BLAST and large genome databases, we can track where and how these genes are spreading. This kind of research is essential for developing global strategies to monitor antibiotic resistance and support public health efforts before these traits become even harder to control.